to the biology class. Today our discussion will be on fate of RBC. We have discussed RBC in detail yesterday, and now everybody knows that RBC lasts for 120 days. And RBC does the main function of carrying O2 and CO2 along with hemoglobin. So RBC is feeding us with oxygen, and each and every cell is getting oxygen and giving out CO2 with this RBC. So RBC is the main components of our blood, our circulatory system. Now what happens after 120 days? We ignore this chapter sometimes. Now let's see how the RBC gets a tragic death. Let us start from here. Once RBC gets aged, that is 120 days or 119 days or something like that, these RBC becomes senile, S-N-I-L-E, senile. That is, it is brittle or old. These RBC are called poikilocytes. P-O-I-K-I-L-O-C-Y-T-E-S. So, these old RBC, old and worn out RBCs are called poikilocytes. What actually it does, this RBC, this is the original shape. They do not remain in that shape. They throw out pseudopodia-like structure here and there, and they become more brittle. So they can be broken. These are called poikilocytes. And ultimately, their pseudopodia is breaks, and they get destroyed. Okay? Now, these RBCs are taken over by RE cells. What is RE cells? Reticuloendothelial cells. RE cells are known as reticuloendothelial cells. They are available in spleen, bone marrow, liver, as well as in the endothelial lining. What is endothelial lining? This is endothelium. Endothelium, these are all flattened cells. So in the endothelial lining, these RE cells are available. Actually, RE cells are of two types: histiocytes and floating histiocytes. These histiocytes, what that does, these histiocytes phagocyte with their phagocytic action. They either in the spleen or the bone marrow or in the liver, say this is RE cells. They engulf the entire RBC. They engulf the entire RBC and crash it. See the depth. They crash the RBC and they become this hemoglobin. That is, once RBC enters the RE cells, they crash. RE cells engulf it and crash the RBC, and then it is known as hemoglobin. Hemoglobin is the vital components of this RBC. What is the capacity of hemoglobin? 100 ml of H2O, 1/3. Say if you put 100 ml of water, it can carry 1/3 ml of O2. At the same time, 100 ml of blood can carry 60 ml of O2. This 60 ml is carried over by hemoglobin. They are doing the best function, and finally we are crashing them to form viral hemoglobin in the spleen. Let it be in the spleen. So in the spleen, now it is easy. I have written systematically. In the spleen, what happens? This viral hemoglobin is differentiated into proteins and heme. They are separated. These proteins are stored or used for further amino acid synthesis. That is, it is a case of protein recyclation. So, in the hemoglobin or in the RBC, whatever protein is there, that gets recycled after this. And the rest remains heme or hemoglobin. This heme, out of this heme contains Fe, that is iron. That iron is stored in the body in the form of ferritin and hemosiderin. So this ferritin and hemosiderin are again recycled for the purpose of creation of new hemoglobin. So our body has the mechanism of recycling, which we don't have in our day-to-day use. We are throwing in plastics and others, it is not being recycled. Here the body itself is recycling all these things. Proteins are recycled as amino acid, and ferritin and hemosiderins are required for further production of hemoglobin. And the rest of the heme is converted to yellow pigment. That is, from RBC, poikilocytes. Poikilocytes are broken down to viral hemoglobin. Viral hemoglobin disintegrates into protein and hemoglobin. Protein side taken care of new proteins, amino acid synthesis, and Fe is stored in the body in the form of ferritin and hemosiderin. And the rest of the heme is converted to yellow pigment, bilirubin and biliverdin. Bilirubin or oxidation produces biliverdin. And some authors are in the view that biliverdin is first produced, which on reduction produces bilirubin. No matter whoever produces early, this bilirubin and biliverdin goes into the blood through the blood vessels. So blood contains RBC, WBC and plasma. This plasma contains globulin, albumin, globulin and etc. etc. So the globulin A1 of plasma mixes with bilirubin and biliverdin. So bilirubin or biliverdin are carried in the form of plasma. Within the plasma they get mixed, and within the plasma globulin A1, say globulin A1 is like this. Globulin A1 in this hand, bilirubin in this hand, biliverdin. They taste this. Okay. Now this bilirubin and biliverdin enters into the plasma of blood vessels, and the blood vessel comes to liver. This liver, within this liver, this compound globulin, albumin, bilirubin, biliverdin complex enters, and globulin A is separated. Clear? This globulin A1, that is which are in the plasma, gets separated. Now what is left? Bilirubin and biliverdin. This bilirubin and biliverdin mixes with UDPG. What is UDPG? Uridine diphosphate gluconate, uridine diphosphate gluconate. They mixes with this, and within the liver, this produces monobilirubin and dibilirubin gluconate. Monobilirubin gluconate and dibilirubin gluconate are produced from this bilirubin and biliverdin. Okay. Now this monobilirubin and dibilirubin enters through the bile duct. Liver has bile duct, common bile duct, pancreas bile duct, and through the bile duct it enters the duodenum of our alimentary system, alimentary canal. So this within this duodenum, this monobilirubin and dibilirubin enters, and it goes to intestine. This is known as, say for example, this is hypothetical intestine. This intestine, with the help of bacteria, this monobilirubin and dibilirubin gluconate is converted to stercobilinogen and urobilinogen. So this is the final product of RBC. Converting here, final product comes here. This, out of this, a portion is reabsorbed and goes out through the form of urine. In the urine, it goes out, and a large portion goes out in the form of feces. FAECES. Feces means stool, and hence the color of the stool. Okay, this is the simplest way of death sentence of RBC. Thank you very much.